So, have you ever wondered how scientists go from just a little vial of DNA to, well, understanding what makes an entire organism tick? It's not just about reading the letters of its genetic code. Nope, it's about turning that code into a map. And in this explainer, we're going to become genomic cartographers, charting this huge, unknown territory inside a genome. All right, let's dive in.
Okay, so picture this. You've just sequenced a brand new organism. Awesome. You now have this massive file with literally billions of letters. A's, T's, C's, and G's. But here's the thing. On its own, that sequence is totally meaningless. It's like being handed a book written in a language you've never seen, with no spaces, no punctuation, not even any chapters. So where do you even start?
And this is really the perfect way to think about it. That raw sequence, it's like having a super high-res satellite image of a whole new planet. I mean, you can see everything, the mountains, the oceans, the deserts, the forests, but you have no idea what any of it is called, what it does, or how it's all connected. The whole process of turning that raw image into a detailed, labeled map, well, that's what we call genome annotation.
Now, this flowchart here, this is basically our roadmap for today. It shows the entire journey, from grabbing a sample all the way to creating this super detailed, specialized map of the genome. We're going to follow this path step by step and see how it's done.
All right, so our first job as cartographers is to survey the terrain. This is all about taking that raw, kind of jumbled data that comes from the DNA sequencer and turning it into a clean, coherent base map that we can actually work with.
So let's look back at our roadmap. We're starting right at the beginning with genome assembly and then prediction of repeats. These are the absolute foundational steps. You know, before we can even think about finding landmarks, we have to piece together the landscape and figure out where all the vast empty spaces are.
So what does that actually mean in practice? Well, first up is genome assembly. You see, DNA sequencers don't give you the whole code in one go. They spit out millions of tiny short fragments. Assembly is this huge computational puzzle of stitching all those little pieces back together into long, continuous strings. Then, once we've got that, we move on to repeat identification, which is basically finding all the parts of the genome that are just the same sequence repeated over and over and over again.
And a great way to think about these genomic repeats is to picture them as the massive deserts or deep oceans on our planet map. They take up a ton of space, right? But they aren't where you're going to find the cities and roads. So by identifying them and then masking them, which is kind of like graying them out on our map, we can focus our search for the really important stuff, the genes, on the landscapes that are much more unique and full of features.
Okay, so our base map is prepped, the deserts are marked out. Now we get to the really exciting part, finding the landmarks. This is the hunt for the functional bits of the genome, the genes themselves.
Right, back to our flowchart. You can see we're following the arrow from repeat prediction straight into this super important box, gene prediction. This is where the real discovery starts. We're not just surveying anymore. We're actively exploring and pinpointing locations that actually do something.
And sticking with our map analogy, gene prediction is like scanning our satellite image to find all the cities, the towns, the roads, the factories. These are the genes, those specific stretches of DNA that hold the instructions for building proteins and running the entire machinery of life.
Now, obviously, this isn't done by just staring at the screen. Scientists use these incredibly powerful bioinformatics software tools. You might hear names like Augustus or Maker, and these programs are trained to recognize the telltale signs of a gene within a string of DNA. They're like a digital toolkit that just scans our whole genomic map and flags all the potential landmarks for us.
Okay, so we found all the cities and roads on our map. Great, but we still don't know their names or what they do. Is this a power plant? Is that a school? That's our next big step, adding the labels. And this process is called functional annotation.
But here's the thing. Annotation isn't just a one-size-fits-all deal. The source material draws this really important line between two types, general and specialized annotation. You can think of it like a two-pass labeling system. First, you do a really fast, automated pass to give everything a basic label. Then, you zoom in on the most interesting parts for a deep dive expert analysis.
And this table here lays out that difference perfectly. On one hand, you have general annotation. It's totally automated. The goal is to just rapidly assign a plausible function to thousands of genes at once by comparing them to these massive public databases of genes we already know about. But then you have specialized annotation. This is slow, it's manual, and it's driven by experts. This is where a scientist with a very specific research question, like, say, what makes this bacteria resistant to antibiotics, will manually investigate just a small handful of genes in extreme detail.
So how does that general automated annotation actually work?
Well, one of the real workhorse tools is something called BLAST. And the best way to think of BLAST is that it's basically a search engine for genomics. You take your new unknown gene sequence, you plug it into BLAST, and it scours these global databases to find the closest known match. So if your gene matches a known gene for, let's say, digesting sugar, you can make a pretty good guess that your new gene probably does the same thing.
Okay, we've built our map. We've found the landmarks. We've added the labels. Now we get the payoff. We can finally step back, look at our completed map, and actually start to read the genome story. And this, right here, is why we do all this work in the first place.
So, the big question is, what's the point? What kind of incredible insights can we actually get from a fully annotated genome map? Well, as it turns out, a whole lot.
I mean, this completed map is an absolute treasure trove of information. Scientists can dig through it to discover genes that produce brand new medicines, like antibiotics. They can find specific enzymes, these things called cazymes, that are absolutely critical for designing better biofuels. We can identify genes that could be targeted for pest control, or just, finally understand the unique biology that lets an organism thrive in its crazy environment.
Let's just take one really powerful example from our source. These things called biosynthetic gene clusters, or BGCs. Think of these as specific neighborhoods on our genomic map, where a whole team of genes works together like a tiny molecular factory. And by using specialized tools, like one called anti-smash, scientists can find these factories, which are famous for producing some incredibly valuable stuff, from the antibiotic penicillin all the way to powerful anti-cancer drugs.
And the story doesn't even end with just one map. A huge part of genomics is about comparison. By comparing our new map to the maps of other species, we can do something called a phylogenetic analysis. And this basically lets us build a family tree of life, so we can see exactly how different species are related and how they've all evolved over millions of years.
So let's just bring this all home. Genome annotation is it's this magical but also really methodical process that transforms a chaotic jumble of billions of letters into a rich, detailed story that we can actually read. It is the difference between a meaningless satellite photo and a detailed map that can literally guide us to brand new discoveries.
And that leaves us with one final, really tantalizing thought. We've only mapped a tiny, tiny fraction of the species on this planet. So you have to wonder, what incredible biological stories, what new medicines, what revolutionary technologies are still out there, waiting to be discovered, hidden in the unread genetic maps of the millions of organisms we share this planet with? The age of exploration, it is far from over.